Steve Minnie, and I'm the Senior Director of Applications for the AFM business in Bruker. And I'd like to personally welcome you to this installment of the Bruker AFM webinar series. This presentation is titled The Power and Diversity of the AFM Pro, presented by Stefan Kamer. Before I introduce Stefan, I'd like to make a few quick logistical comments. First, we encourage your participation during the webinar, and if you have a question, please type it into the questions dialog box on your screen. We'll accumulate these questions through the presentation, group them together, and then answer them at the end of the prepared remarks. Quite often we have more questions than we can answer in the session, and in those cases, we'll be sure to follow up with answers via email afterwards. Also, if you'd like to review the presentation or pass it on to a colleague, the webinar will be posted online, usually within a week, in the webinar section of the Bruker webpage. Also, as a follow-up to this presentation, you'll be receiving an email with this link. Finally, when you exit the webinar, the software will ask you to take a brief survey, and we would very much appreciate it if you could take the time to complete this, as it helps us pick the topics that are most important to you and generally make the series better. So let's get started, and let me introduce our presenter, Stefan Kamer. Stefan earned his master's in chemistry and his PhD in physical chemistry in 1992 from the Technical University of Braunschweig in the group of Professor Lachmann. He started working in AFM as a scientist for topometrics in Europe in 1993. Before coming to the U.S. in 1995, Stefan lived in Japan working as a guest scientist for Hitachi in the field of near-field optics and phase change materials. Since 1995, Stefan has worked for Mica in various technical and management and product marketing roles. Currently, Stefan is a senior member of the Bruker Applications Development staff in Santa Barbara, California. So now let me turn it over to Stefan for his talk titled The Power and Diversity of the AFM Pro.
Yeah, good morning and welcome everybody on this early morning here in Santa Barbara. And let's get started with our presentation today. So AFM is a very powerful and diverse tool, and what actually makes it so diverse is the ability to use a variety of different probe tips. And these probe tips are the key to unlock this diversity, because they allow us to measure a quantity. And that quantity that we can measure here is based on what interaction that specific selected tip is sensitive to. And these interactions then can, of course, vary. We can measure surface topography, probably the mostly used application for an AFM to date. We can measure molecular forces and then go into the field of pulling or molecular recognition. We can gather these days nanomechanical information with nanometer resolution by using peak force QNM. We can gather a variety of electrical information, conductive capacitance, surface potential, and so forth. We can combine AFM by using the tip to gather optical information on a nanometer scale, and then get Raman, infrared, or fluorescence information out of the AFM. And of course, we can get thermal information or calorimetric information from our AFM if we choose the correct tip. And choosing the correct tip is basically what started us to give this webinar here, and that's what we are going to talk about. So what do we cover in this talk? We can't, of course, cover all the topics that I just mentioned, so we have to concentrate in this webinar on a few here. And in order to get quality information, one needs the right tools. One needs a high-performance AFM, and one needs the correct tip. And we're going to cover the correct tip in order to get high-performance, high-quality topographic information with as high resolution as possible. And I'm going to have 3 examples here. Information of a single molecule, DNA, in fluid and tapping mode, an S layer of an E. coli bacteria membrane, showing the 40 nanometer periodicity in fluid, and a contact mode image also obtained in fluids on human endothelial cells. So what kind of tips do we need to get data like that, and why do we need these tips? This will be the topic of today's webinar. So we have to step back a tiny little bit, and I'm not going to go into great detail here. So what we do in most AFMs these days is we use a beam-bound system, and when we talk about tip, of course, we're always a little bit sloppy, because what we really talk about is the assembly consisting of a cantilever and a tip at the same time. So every time when I say tip, I basically mean the tip cantilever assembly, and that is very common when you go through the literature here. So just briefly, in the beam-bound AFM, what we have is we have light coming from a diode or superluminescent diode or from a laser. It's focused on the backside of a cantilever and then bounces back into a photodetector, and the movement of the cantilever is amplified so that you get an amplified movement of the photodetector, and then you read the photodetector out to get information about what your cantilever is actually doing in space. Now it's important to note that the beam bounce is often also called a light lever effect, because we're leveraging the movement of the cantilever basically by the distance between the detector and the cantilever divided by the length of the cantilever. However, it is important to note here that the ultimate sensitivity is independent of L to a great extent. I mean, you get a little bit of, of course a slight benefit by making your cantilever shorter, because you get a larger movement for a certain angular displacement of that cantilever. But the ultimate sensitivity can be shown to be independent of L, but rather proportional to 1 over S. And that's why if you really want to use super small cantilevers, you need a different AFM than you may be used to. Now, where do we find information about tips? You can go to Bruker AFMprobes.com, and there are a variety of information. You can either type in information about a specific probe that you're interested in, or you can look at suggested probes based on the instrument that you're using or the application that you're interested in. A quite useful site, check it out. There's also a probes catalog electronically available that you can download and take into the lab to always have these information handy. Now, when you go to the probes website, you often find like a table like the one I show you here. On the left side, we see sample types. You can see here it goes from biomolecules over cells to tissue samples. Then has a variety of probes. Probes are typically made from either silicon, based on standard semiconductor processes, or from silicon nitride. So hence you see these two materials showing up here. And then you have these rather cryptic names for the probes, O-TESPA, R-TESPA, DNP, and so forth. And then on the right, you see our suggestion on which probe to use in either liquid or air, and then for which application. And we have tapping, contact, and force curves here. And we're now going to pick a certain area. We're going to pick force curves, and I circled the area in red, and our suggestion here is something like biolevers, SNL, MSNL probes. And we're going to look why do we select these probes and what we can achieve with these probes. Okay. So what's a force measurement? AFM is often used for force measurement, and it's a basic operation, the most basic operation of an AFM, as a matter of fact. And it generates plots like the one that is shown here. Here you see the tip deflection on the vertical axis plotted against the tip sample distance, and on the when we look at the orange curves here, that basically shows a tip sample coming close together, and it doesn't really matter for that purpose here if you bring the sample close to the tip or the tip close to the sample. We see we bring these two objects together. At one point the tip will contact the sample, and then it will deflect with further movement of the two objects. When we pull them back, we're going to have some hysteresis, and at one point the cantilever leaves the surface again. And curves like that can give us a variety of mechanical information, compliance, elasticity, but it can also give us information about adhesive forces between the tip and the sample. Hooke's law describes these force measurements pretty accurately, meaning that the force measured is proportional to the cantilever spring constant and of course the movement of the sample or the cantilever. So when we look at that, so we basically already have a very simple solution. So if you want to make our instrument sensible to measure really small forces, I mean, we just make a super soft cantilever and have a go. Or is that not true? There's a nasty effect that comes into play, unfortunately, and that is called noise. And it's actually interesting when people talk about noise, because people go to great length of looking at the noise of the AFM system. Typically before they go shopping, but there's also a lot of talk about noise once people have the AFM. But what noise do I actually have to look out for when I want to do these force measurements? So there are basically two types. There's noise that is related to your AFM system in itself, and then there's of course noise that is related to the cantilever that you choose. So when we talk about noise in your AFM, probably what you want to look at is deflection noise, and that is of course an instrument-specific measurement. But with our Bruker AFMs, it's typically below 0.1 nanometer or 1 angstrom. So given that you can have a cantilever with a spring constant of, let's say, 10 piconewton per nanometer, you should be well able to measure forces below 1 piconewton if you just apply Hooke's law here. So, however, there is something that is often overlooked, and that is the thermal noise of the cantilever. Where does that come from? It basically comes from the fact that nothing stands still, so to speak. Statistical thermodynamics led to the description of Brownian motion, and Einstein formalized that in 1905. And that basically means that your diffusion constant, diffusion is proportional to a constant times the absolute temperature here. When we now apply that to a cantilever rather than some particles, let's say, in solution, we can apply the equipartition theorem that relates the temperature of the system with its average energies, and we can do that by putting on the left side here the mean squared amplitude of the cantilever motion and equal that to each degree of freedom at t, expressed as half over k b times t, and then we arrive at an estimation for the RMS noise and force measurements. And that noise will, if it's bigger than your instrumental noise, of course determine the ultimate resolution you can get from force measurements. And when we take an example here again from the Bruker website, we pick the OBL lever, often also referred to as the bio lever. We can simply plug the spring constant in here, and we arrive at an ultimate resolution of 5 piconewton that we can achieve with that cantilever. So you will not be able to go into the femtonewton or even lower range. I mean, that is a limit here that is given by the cantilever that you choose. So you can see that the AFM in this specific case is absolutely not the limiting factor, taking of course that you start with a good AFM in the first place, but it is actually your cantilever that limits your ultimate force resolution. Now, what can you actually do once you know that? You can do a bunch of interesting things. You can, of course, do simple force curves, but you can also modify your cantilever, and a lot of people do that, and then basically add chemical specificity to your force measurements. And you can do that by simply modifying the surface of the tip. And there's a lot of chemistry available to modify nitrides, and even more chemistry to modify silicon tips. And if you modify these tips, then suddenly you can go ahead and do things like molecular recognition mapping. I'm taking a step ahead here because here I show basically data not based on single force curves, but I extend it by using our new Q&M mode to just go into mapping right away. So what are we looking at here? We're looking on the left side of a topographic image of malaria-infected cells, and on these cells, it's believed that these little knobs, which you might see in the right of the red images on the top, are believed in the adherence to endothelial cells, and they prevent the cells from going into the spleen, which would then actually help issue a kill order for these cells. So because these cells never arrive there, they can't be killed that easily. So how do we verify that these knobs are really responsible, that these are really the adhesion centers? Well, it's a quite straightforward experiment. We take a probe and we functionalize the probe with a specific receptor for these centers. And then we just go ahead and do our force measurements, and what we get is shown at the bottom. On the left side we have an adhesion map, and on the right side we have a topographic map. And you can clearly see that every time when you see an increase in adhesion, you can relate it to a knobby structure in the topography. Now, skeptics might say immediately, "Oh, well, it's just an artifact," because when you see a knob in the topographic image, you get a bump in adhesion. That's a valid argument. However, you will notice there's a little blue arrow in the topographic image pointing to one topographic bump here. There's something on the cell which is clearly elevated to about the same height as all the other topographic features, but it clearly shows no adhesion in the adhesion image, meaning that it's a great indication that our adhesion map is really based on the proper interaction that we were looking at here. If you want to go ahead and of course do single force curves, you can still do that and verify the measurement, but it's just very convenient to do these mapping information because you don't have to go in by hand and create these maps and maybe accidentally miss an object. Let's talk about miss an object. In the next slide here, we show again a property map, but this time based on an assay of live and dead cells. So what we have on the top left here is a fluorescently labeled assay of live and dead cells. The live cells are labeled with a dye in the green, and the dead cells are labeled with a dye fluorescing in the red. And we can use that fluorescent image in our catalyst system to simply, without scanning the image, just take that fluorescent image and move the probe to a number of locations indicated by these crosshairs here. And you can see there are a bunch of force curves locations selected on the live, on the dead cells, and on the substrate, which is not hard in this case, but more like a gel-like structure. You can see the corresponding force curves at the bottom. And you can see that the dead cells definitely show a big difference in elasticity as compared to the live cells and the substrate in this specific case. An interesting point is now we can go ahead and again do the peak force imaging. Just do a quick peak force map, and you can actually see that you see a very similar information in the peak force deformation channel at this point. That's a channel that gives you information on how much deformation you can achieve under your imaging conditions on a certain sample. And you can see that on the two live cells at the bottom, you have pretty much uniform deformation going on, whereas on the dead cell on the top, you have a variety of, you have a big variation in deformation going on, and also shows a quite significant higher deformation. But what is even more interesting, that you see another dead cell which shows up on the top, and that cell was not visible in the fluorescence image. That's I think a great example why this mapping based on peak force tapping is often more useful than to do a single force curve, because if you would have just done the single force curve, you would have completely missed that cell. Whereas here we don't miss it, and now we can go ahead and if you want to analyze it in greater detail. So imaging. So now going from force curves to and contact mode to imaging here. For imaging, we would like to talk about a tapping mode here, and in air you can see there a bunch of silicon probes suggested, OTESPA, RTSP or RTSP-A, and by the way, the A always stands for aluminum coating, and I strongly suggest, and like years ago, I would have never used an aluminum coated probe because the coating was really not very good, and there was often something on the front end of the probe. These days, the coating is really good. It's only on the back side of the probe. It increases your signal, and there's really no reason not to use an aluminum coated probe. So go ahead and do that. And at the bottom, you see that in liquid environment, there again the same probes actually selected that we used for force measurement. So rather interesting fact here. We also want to keep in mind that the resonance frequency of a probe drops significantly when we move the probe from air into fluids, and so does the Q factor. And we're going to look into this. So what do we do in tapping mode? So in tapping mode, we don't use a static cantilever. We modulate our cantilever often by means of a small piezo that's mounted to it, and we operate it at resonance. And if we just have a small force acting between the tip and the sample, we can simply use a harmonic oscillator model to describe that motion, and we can see on the right side, if we do so, then we end up basically with an effective spring constant of our cantilever, and it's basically determined by force derivatives. So that's one thing to take away here. When we do tapping mode, we don't look at a force per se. We're always dealing with a force derivative, so df to dx, x being the distance here. When we come closer to the sample, that force basically changes our system. The stiffness changes, and we use an effective resonance frequency in this case to operate our feedback system. So where on a force curve do we actually operate when we do tapping mode? And this is an object that a drawing here that can help us understand that. So we have here force plotted in the vertical direction versus distance, and in blue we plotted basically the repulsive Coulomb interaction, and in green the attractive van der Waals interaction, and the corresponding force curve here in red. And tapping mode is basically making use of trying to get really close to the surface, making use of the repulsive Coulomb interaction. And already from that curve here, we can see that repulsive forces is really where you want to be when you do, well, if your goal is high resolution imaging, then your goal should be to use the short range Coulomb interaction, because you have a much shorter range, which will then result in higher resolution than if you would use the attractive forces, because the interaction distance is much larger. So talking about lateral resolution, what is it actually? How do we have to deal with it in scanning probe microscopy? So it's basically the smallest feature that you can resolve, and in optics, it's determined basically by the spot size of your focus beam. If you don't try to play any funny games here, in SPM, it's and we take that up front. It's determined by the intuitively you would expect that it's determined by the tip size, and of course by the tip sample distance. Now, it's of course not as easy as it seems, because what does it really mean, tip size? It depends on what does your sample actually see of a tip, because when we just look at a tip and we look at the drawings of a tip on the Bruker AFM probe website, I mean, these tips are rather large. So how do we get actually nanometer resolution? So how much of the tip does the sample actually see? And you often hear that along the same line that, in SDM we use these super sharp tips, which is actually not true. In SDM you can use a rather dull tip that you just prepared by using your good old nail clippers. The thing is that the sample only sees a very small portion of the tip because of the exponential tip sample dependence here. So in force microscopy we have to therefore look at the change in tip sample interaction forces in order to be able to define a little bit better lateral resolution. That brings us to a point, why do we actually want to stay close to the surface, as I mentioned earlier already. Let's take a very simple example here of electrostatic measurements. We can take a grounded tip here away from a point charge Q, as that's shown on the left, where we see Z plotted against distance here. We see the tip of a radius A distance S away from a surface, and we at a distance X we see a point charge Q. And it's rather obvious that the force on the tip will of course be a function of that distance X, with the maximum being at X0, so right over the point charge. Now let's take a second point charge, and we can actually show that the force is a function of X, S, and R. And if we go through the formalism here and peel that apart, and I spare you that, we can actually look at the bottom picture here, where we plot the force versus X again. We have these two point charges, Q1 and Q2, shown here, and the resulting force that the tip sees. And so delta lat is now the lateral resolution. That is the resolution that that we can achieve if you can still separate these two charges. We can show that this charge is the lateral resolution is proportional to the square root of the tip radius, but more importantly, it is directly proportional to the tip sample distance S. So making the tip sample distance actually more important than the actual size of the AFM tip that we're using here. And that is, in a very simple way of looking at it, that is the reason why you actually want to stay close to the surface and not far away like you do in non-contact mode for the highest lateral resolution, if that is your goal, which it is for the most of us. So now what do we have to do in order to stay close to the sample? And that brings us then to the answer of what kind of tip we have to use. So what we want to do is we want to stay close, but at the same time we want to avoid the instability that the jump into contact would cause. Now, how do we do that? So when we look at the left side here, we have our tip above a sample, and we have of course opposing forces acting on that assembly. We have the force defined by the spring constant of the cantilever pulling in one direction, and of course we have the tip sample forces, if they're attractive, pulling into the other direction, and our total force is a combination of these two. And what we want to achieve is shown on the right side, where we plot tip sample distance with S being 0, the tip on the surface here versus time. So what we want to do is, when we ramp our tip slowly towards the surface, we want to have a linear relation here. What we want to avoid is what's happening at here F total equals 0, that we have an instability and the tip surface forces overcome the restoring force here, and our tip snaps instantaneously to the surface. So in order to achieve that, we need a tip with a high enough spring constant, and hence we selected these tips that we shown in the table in the beginning, which have a spring constant of around 40, 50 or so Newton meters, which are high enough to avoid this jump into contact here. You can always calculate spring constant. And I show you a formula at the bottom here with the modulus of silicon plotted. So simply plot your dimensions of the width. If the width on the top of the bottom is the same, of course, just use it, and then it's basically proportional to the power of 3 to the thickness of the cantilever, and inverse proportional to the length to the power 3 of the cantilever. So the shorter the cantilever, the stiffer you get. Let's look at the whole thing from a different perspective. So when we do tapping mode, we plot here our cantilever versus frequency. We work at resonance, as we already established a few slides ahead. So and then of course the force we can actually work at is somewhat proportional to what kind of amplitude we're going to select as a set point here. So we start with a free amplitude of X nanometer, and then say we want to have it reduced to Y nanometer. So that difference is basically one of the factors that determines our actual force. And then of course it's dependent on the spring constant, and we just established that we need a quite stiff tip, so a high K to avoid that jump into contact. And then of course it helps a little bit by using a high Q here, but we really don't have any influence of the Q. That's something that's given by the design of that lever in this case. And then we end up at some forces in somewhere in the piconewton range. We can't go infinitesimal fast because we have a resonating system here, and that means we have some kind of time constant tau that defines our response time that we have when the cantilever encounters a change here. So the higher the Q, the higher that recovery time is. But we have another problem. If we have a high energy system and we can't pump an infinite amount of energy as quickly as we want into it, because we are basically held down by that Q again. So we have to wait like Q before our cantilever actually responds to that. And I showed the formula here at the bottom. And one thing we have not talked about up to now is that when you work in air, you have often an adhesion layer on your surface. I mean, the moment you have a hydrophilic surface, I mean, you have a strong adhesion on your sample. And we're using our tip cantilever here to actually establish feedback. So meaning our tip cannot get sucked into this adhesion layer. It has to have enough energy to get out of the adhesion layer at each cycle. Otherwise, the whole feedback would collapse. And if you make a simple estimation that it takes about 10 to the minus 18 or so joule per cycle to leave that adhesion layer, then you can actually see that with an amplitude of 20 nanometer, a K of 1 Newton per meter, and a set point of 0.9, I mean, you will not be able to do so. So you need a little bit more energy, and that of course is the limit here in what kind of force resolution we can achieve with our specific system in tapping mode. Is there a solution for it? Of course there is one, otherwise I wouldn't show you that slide here. And the solution is quite simple. Actually, when you look at it, I mean, it took years to come up with it, but it is rather simple. And the solution is to work off resonance, to work sub-resonance. And because then again, our force here is determined by Hooke's law, and we talked about that in the beginning. And then we can actually use a way softer cantilever. And we have seen that these cantilevers are great for very sensitive force measurements, just for point measurements, but they should also be great for imaging if we just use them in the correct mode. And that is a sub-resonance mode, and that's the mode we call peak force tapping. And the trick here is that we do not use the cantilever in itself to establish the feedback condition as we use in tapping mode. We separate effects here because we use the Z piezo to move the cantilever up and down and pull it free of the adhesion layer. The Z piezo has infinite stiffness, infinite power for our specific application here. So by separating effects here, we can use actually a very soft cantilever and then gain a significant amount of force resolution. So how does peak force tapping work? I just show briefly a diagram here. Again, we move our tip up and down, and that is similar to tapping mode, but we don't use it at resonance. We use it at sub-resonance, and we can monitor the deflection of that tip. And when the deflection of the tip here reaches a certain set point, we simply stop our motion and reverse it. And again, because we use the Z piezo to pull the tip free of the adhesion layer, we can use a very, very soft cantilever and gain a significant amount of force resolution here, so be much gentler to our sample than we could ever be in tapping mode, because we don't have to deal again with the adhesion layer. I mean, we still deal with it, but we use the Z piezo to deal with it instead of having our cantilever having to deal with it. So what kind of resolution can we now expect that we looked at tips here? We have some examples of alkynes here on graphite, and in the past, when we got like images of the C60 alkane here with a chain-to-chain distance of on 40 nanometers, we were actually quite happy. And then we improved, the tips got improved, methods for do tapping mode got improved, and people started to get images of C36, which are now a much shorter distance of 4.5 nanometers between lamella here. We were really happy, but now we can routinely get images of a even shorter alkane here, which a distance between lamella of only 2 nanometers. And in order to get a 2 nanometer distance between lamella, you need a tip of around a 1 nanometer diameter, and of course you need a quite low force in the nanonewton, 1 nanonewton range or so, in order to do that reproducibly without destroying these lamella. And these images were done with peak force tapping in air. That's something that is again routinely doable. And in air is where tapping mode actually has its issues for imaging, because you can really not get lower in force resolution. However, tapping mode is extremely useful and shows, of course, great strides still when you use it in fluids. And in fluids, a lot of the effects we talked about are eliminated. And I show you some absolutely stunning data here, obtained in fluids in the group of Frieda Mugele, together with Mr. Ebeling at the University of Twente in the Netherlands. And they imaged on a just regular multimode with a regular fluid cell, clay-like materials, and Gibbsite is one of these. And on the left we see here a 1x1 micron overview in fluids of these of that sample. You can see it forms these platelets on a mica surface. They're about 10 nanometers in height. And most people were happy with getting an image like that. But what these guys did now is like, hey, let's see how far we can push it. And that's shown on the right side. And what you see here is an atomically resolved image of the surface of these Gibbsite platelets. You see the topographic information on the left and the phase image on the right side. What you can see is that we see that brief part that we see in the image here shows 3 different terraces. You can clearly distinguish the terraces. You can see the energy at the step is higher. That's why you see a bunch of particles adhere to the step edges. And when you look really careful, you can see that the atomic lattice is clearly resolved on actually all 3 of the terraces, because of the color depth here you cannot see. And it's hard to see it on the screen here, that even in the lower terrace you can see in the phase image the terraces are clearly resolved. What is even more astonishing here is not only do you get the resolved lattice, but when you look carefully, and I indicated that area with the blue arrow here, you can actually see a single molecular defect. That is a clear indication that not only do you get lattice resolution here, you get true atomic resolution with an AFM in liquids. And not only is it with an AFM in liquids, it's with a regular multimode that probably 70% of all your listeners are using right now. The only thing that these guys did is they used a fast scan B cantilever, which is a cantilever that we normally advocate on use for our fast scan system. But it's not really a super short cantilever, and it's not a super expensive $360 or so cantilever. It's a regular silicon cantilever that you can buy. Just use it on your multimode, give it a shot. I mean, you can see what's possible doing with it. So then, of course, if you image these platelets, I mean, of course, you get curious and see, hey, what happens if I go to the substrate? And that's what these guys did here too. And now they also compared tapping mode to contact mode, and contact mode is shown on the right side. And here you get the images that you may be used to in AFM. You get your letters, your Mica letters in this case, clearly resolved, but you do not see any single molecular defects, because the interaction area between the tip of the surface is just too large. However, when you switch to tapping mode, you can again clearly see the letters as indicated by the blue dots here, but you can also see that single molecular defect indicated by the blue arrow here. Again, an indication that you get true atomic resolution and not just lattice resolution. And again, taken on a regular, plain vanilla multimode AFM from Bruker. So now that we have deciphered basically what is necessary to do very sensitive force measurements and high resolution imaging, we can conclude our talk here by saying that if you choose the right cantilever for the job, you will fully unlock the potential of your AFM. It's often beneficial to make some rough estimates. They might not be 100% accurate, but it doesn't matter. Make some rough estimates what can be achieved with a given cantilever, and hopefully I've given you some ideas on and some tools on what you can do there. Peak force tapping achieves extremely high resolution in air due to the superior force control and the ability to use soft cantilevers as compared to a conventional tapping mode. However, by working in fluids, tapping mode using small amplitudes in this case can produce a true atomic resolution data. And again, don't believe the hype that you have to buy a new gadget in your toolkit. I mean, you can do very well with your regular existing multimode AFM in this case here. I would like to thank my colleagues Andrea Slave, Stephen Minnie, James Shaw, and Chanmin Su for helpful discussions to make this webinar happen. Thank you.
All right, thank you very much, Stefan. We have a number of questions. I'll try to get to them all and group them together when possible so we can get through as many as possible. If you have a question, please type it into your questions dialog box. That is the way to enter questions in so that we can see them and read them. There's another option of raising your hand, which doesn't give you the option of typing in the question, so please don't select that option, but go to the questions box and type in your question. So there's a number of questions on the benefits of low noise, and I think it sort of came at the time when you're on slide 10 on the OTSP, and then also on the peak force tapping versus tapping. And so the essence of these questions are, what is the benefit? And these may have come before some of your later data sets, but what is the benefit of getting down to, for example, 5 piconewtons noise that you showed for the OTSP?
So the 5 piconewtons of noise were shown for the ONL, for the biolevers on the force measurements. Of course, when you do force measurements, you want to often measure small forces because you want to, for example, unravel a molecule. And you want to figure out what kind of forces occur during unraveling these molecules. And if you use a very stiff cantilever for that case, of course you get a beautiful looking force curve, but that cantilever simply will not deflect at the small forces that are required to do these measurements. Hence, you want to use a very soft cantilever. Now we can, of course, make the same argument for imaging, and that's what I tried to do here by introducing you to peak force tapping, because we can use that same soft cantilever to do imaging now, and then gain the same benefit of being sensitive to very small molecular interactions between the tip and the cantilever. And again, you have to separate the force resolution here that is possible between your instrument and your cantilever. That's another important point here to make. And typically the sensitivity of your AFM is much better than that of the cantilever anyway. As long as you start off with a good AFM, and I step ahead here and say that we make good AFMs that are good enough. So you don't have to worry about that. I mean, if you take a multimode, for example, the noise you have on that multimode is sufficient to do measurements like the ones I've shown here. And I mean, again, you can believe me, or you can just believe the data. And data usually do not lie. And I've shown you the data here.
So along the cantilever and sort of the PFT combinations, question is, won't soft cantilevers in peak force tapping cause problematic ringing in the force curve?
Of course, cantilevers can cause ringing in the force curve. I mean, so does a tapping cantilever when you hit an object. I mean, and that's why I showed you that the time constant tau there, that you have to basically wait for ringing to stop. I mean, right? When you look at the imaging as we perform it when we do peak force tapping, you will actually see when you look at some real-time force curves, you will see some ringing. That's unavoidable. However, and that's where the trick comes into play. You have to make your software that analyzes the motion of and the behavior of the cantilever smart enough to ignore that ringing. And that's exactly what we do. So even when we have a cantilever that rings, we're still perfectly able to extract the peak force out of that signal. So will it ring? Yes. Does it affect our ability to do imaging at extremely low forces? Absolutely not.
Okay. I guess in the last in that group, Ms. maybe summarize, explain the trade-off between the low spring constant peak force tapping probe versus the high spring constant tapping probe in terms of other aspects of performance. So I guess that perhaps would be speed, lateral resolution, ultimate force control, just general performance. What are the other trade-offs?
Well, in ultimate force control, it's so there are two things, and that's, I think there's a common mistake that we make when we simply compare the probes. The trick is here that these modes are completely different. First off, you can use a stiff probe in peak force tapping. Nobody prevents you from using a tapping mode probe in peak force tapping. And we actually do that if we want to exert a really high force to the sample, as we want to do, for example, if we want to apply a high force to make a mechanical measurement into like a piece of silicon or some stiff sample. If you want to make, for example, a nanomechanical measurement, you have to indent your sample. If you don't indent your sample, you cannot make a nanomechanical measurement, right? So you have to separate probe and mode here, and if we do that, hopefully I've done that here, then you will see that I couldn't go into great detail, of course. Then you will see that in peak force tapping, you're actually able to directly measure a force. You can measure a force at each single tip to sample contact. In tapping mode, you do not do that. In tapping mode, first of all, you don't measure a force. You're measuring you're always looking at a derivative, right? You're looking at the change of force with distance, for one. Second, you're averaging. Typically, your tapping mode signal goes into a lock-in amplifier, and out of that lock-in amplifier comes a signal, and that signal is what you use for tapping mode. So what comes out of your lock-in amplifier is not the signal. Is not the signal from a single tap. It's the average of over a large number of taps, depending on what time constant you set on your lock-in amplifier. So not only are you not able to look at a force directly, you're also not looking at a single tap because you're averaging at the same time. So, and you have to separate these things from using a tip. And the other difference is that in tapping mode, you're using the cantilever also to establish feedback. If your cantilever gets sucked in by the adhesion layer, it's game over, right? And you can have a hydrophilic surface and say, oh well, I can use really small amplitudes to keep my force down. And I showed you the formula. Of course, if you keep your amplitude down, I mean your force is down. However, if you have strong adhesion, I mean you just need one patch of strong adhesion. I mean then it's game over, and your cantilever gets sucked into the surface. Whereas in peak force tapping, we separate these issues and use the Z piezo to move the cantilever without affecting feedback. And these are the two things I think you have to separate and look at in their own context.
That maybe is a good segue. There's a sort of a large group of questions on focusing on really the apex of the probe, and I guess to group two of them together, it is what causes tip wear in tapping mode, and another question, what probe should I use if I want to minimize tip wear?
Tip wear is of course caused by tip wear can be caused by a few things. I mean, one thing is of course something gets messed up and your tip makes hard contact with the sample, and then either your tip or the sample gives, and something breaks off your tip, and then your tip's not as sharp as you started. As a matter of fact, it's interesting to look at in tapping mode is often the approach. I mean that does it to you. So at the very first tap to the surface snaps off a small part of your tip. So what you want to do is you want to try to minimize the amplitude. Obviously, that keeps the force down. You want to make your intelligent algorithm your approach algorithm as intelligent as possible, and we have a lot of intelligence built into our approach algorithms. You don't even have to think about it. The system is supposed to do these things for you, but keep the amplitude low. But again, I mean, if you have a lot of adhesion, you can't do that. You have to keep it at a certain level in order to be able to break free of the surface, not to get sucked into. And of course, silicon tips are the tips you basically have to use because you need that spring constant to avoid the jump into contact in tapping mode. And of course, do not scan too fast, because if you scan too fast, I mean, you run into the issue that I showed you the time constant there. You need a certain amount of time to establish clean feedback so that you can really avoid accidental tip sample contact here. Yeah, and then of course the answer would be just use peak force tapping if you have the possibility to do so, because again here you can use really soft cantilevers. So even when you even if it would happen that you have like somebody bumps into accidentally a little bit too hard into a feature, you have a really soft cantilever, so the force you exert is significantly lower for a cantilever with a 0.1 Newton-meter spring constant than for a cantilever with a 40 Newton-meter spring constant. So even if something happens, I mean, the effect is much less. So that would be my advice here.
Okay, so keeping with the apex, two questions on how do you clean a probe? Does Bruker have a standard procedure to clean tips? Do we need to clean a new tip for best results? And again, how do you clean a probe if something is sticking to it after multiple uses?
Yeah, I don't think I'm a great expert on cleaning probes. I never do. I have Steve sitting here, but I have to admit that I usually throw my probes away if they are dirty. He's the one that signs off the budget, so that I maybe shouldn't say that. But there are ways to clean probes, and in fluids you can people do that. And the data I showed you from the group in 20, I mean, they definitely cleaned the probe with alcohol. I think it was alcohol, if I remember that correctly, before they used it. There are a variety of cleaning methods, and I think we have an application note on it. If I'm not completely mistaken, it might be in one of the bio application notes. If you contact me offline, I can look it up for you. if something sticks to the tip, I would usually say that's game over. You basically do not get rid of it at all. That's one of my pet peeves is when I see people, when you calibrate an AFM, we typically use a calibration grid. And one of my pet peeves is I absolutely cannot stand dirty calibration grids, and there's so many of them around, because what happens is each time you scan over a piece of dirt, I mean, it will stick to your tip and it will completely mess up not only your tip but also your measurements. So one advice would be try to keep your samples clean if you can. Try to avoid loose stuff on your samples. That will inevitably kill you. I mean, your tip will pick these loose particles up. There's just nothing you can do about it. Try to avoid static electricity on samples if you can. I mean, that will also have an effect on how much your tip can pick up. Try to use a little alpha source that you can buy in a photo supply store. So that you can buy these little brushes, which when you open them up, there's a little polonium source in it. Just stick it next to your sample to avoid static electricity. These things, but can you clean particles off that are at your tip? I mean, in my experience, you cannot.
Okay. So going back to the red blood cell data, how do you calculate the pull-off force when using the functionalized probe?
Well, you calculate the force using Hooke's law. I mean, it's like I showed you the force curve at the bottom. So basically, the difference between your baseline, so basically when the cantilever is not deflected, and the lowest point before the cantilever leaves the surface is the adhesion force here. And if you know the spring constant, which you can of course calibrate and should calibrate if you want to know the exact force, then you can easily calculate the force here that was required to overcome that adhesion.
Okay, so we're scheduled to end at 9 o'clock, so I think we probably have time for one more question. There are quite a few more questions on the list, and if you did ask a question, we will be sure to get you an answer offline. So for our last question, I think this is perhaps a good one to wrap up with. You spoke mostly about biosamples. What would be your rule of thumb / formula to select a tip for polymers, and then especially conducting polymers, and then sort of a follow-on to that, would you compromise on softness through length or thickness, or what's your rule of thumb?
Basically, whatever applied here for bio would apply for polymers too. However, with polymers, there's often what you want to do is the top layer, not just of a polymer. The famous physicist at one point said the devil made the surface. So your surface is always different than the bulk. And what you often want to get in a polymer is you want to get information about the bulk. So the only way you get that is if you actually indent your polymer slightly so that you get actually information from the underlying layers here. So what you can do then, if you use tapping mode as an example here, you use like instead of maybe a TESP layer lever, use a little bit softer and longer lever, use an FESP lever that allows you actually to push a little bit further onto that surface to get information from the underlying layers, but at the same time it's not as stiff as a TESP layer, so there's you can still get away without too much damaging effects here.
Okay. Well, I'd like to thank Stefan again for a very insightful presentation, and thank all of you for attending. Again, please take the exit survey on your way out, and we will be following up this webinar with an email to a link where you can download a copy of this webinar to watch it again or forward it to a colleague or get the slides. And then also please be on the lookout for our next announcement for our webinar next month, and we hope to see you then. Thank you very much, and goodbye.